Hi everyone, and welcome to my video on pharmacodynamics. You know, you can give a drug to a person and have dramatic results, and then you can give the same drug to a different person with very little effect. If you remember what we learned about in pharmacokinetics, we can see how the mechanism of action and the side effects can be somewhat predictable depending on the person you're giving the drug to. If you remember that pharmacokinetics is what our body does to the drug. But regardless of the variability in the person that we're giving the drug to, sometimes the responses are not as easily predicted. That's where pharmacodynamics comes in. Pharmacodynamics comes from two root words, pharma meaning medicine or drugs, and dynamics meaning change. So in essence, pharmacodynamics means how the drug changes our body. So once again, pharmacokinetics is what our bodies do to a drug, while pharmacodynamics is what the drug does to our bodies. Your textbook elaborates on this definition, stating that pharmacodynamics is the branch of pharmacology concerned with the mechanism of drug action and the relationship between drug concentration and the response in the body. So why is pharmacodynamics so important? Well, knowing the principles of pharmacodynamics will provide you as the nurse with the ability to predict if a drug will have a particular response in a patient or whether a dose will be effective or safe in any given client. A frequency distribution curve is used as a graphical representation of the inter-individual variability in a drug response among clients. Inter-individual variability refers to the differences between the responses in individuals who took the drug. Let's look at a frequency distribution curve. We can see that the vertical axis is the number of patients, while the horizontal axis is the dose that was given to elicit a response. Notice that a few of the patients responded at low doses. As the dose increased, more and more patients responded, while some of the patients required higher doses of the drug to elicit a response. The peak of the curve shows the largest number of patients responding to the drug. We don't get the magnitude of the response here, just the number of patients. The middle value of the frequency distribution represents the median effective dose. That indicates that it takes effective dose ED to produce a response in 50% of patients. Another word for effective dose is what is called the average or standard dose. Based on the findings, it is important for the nurse to note that we can predict that only half of the population here will experience a therapeutic response from the drug. That means that some clients will require more or less of that drug. It can also mean that some clients may experience toxicity from a drug or standard dose. What does this mean to the nurse? It means that the skill of the nurse to observe the client, take vital signs, and monitor associated lab work is critical in ensuring the client's well-being. So you can see how it's not enough to simply memorize drugs and their average dose. There's always a bigger picture. In order to ensure safe pharmacotherapy in humans, preclinical trials as part of the drug development process will often determine the median lethal dose, or LD50, in animals. That is a lethal dose in 50% of animals tested, usually mice or rats. That lethal dose, 50 in animals, is then compared to the effective dose 50 in humans. Shown on a frequency distribution curve, you can see that, in this graph, 10 milligrams of drug X is the average effective dose, and that 40 milligrams is the average lethal dose. Both the effective dose 50 and the lethal dose 50 are used to calculate what's called the therapeutic index of a drug. So the larger the difference between the two doses, the greater the therapeutic index. So in this figure, the ratio of a drug's lethal dose 50 to its effective dose 50 is 40 divided by 10 to equal 4. Therefore, it would take a magnitude of approximately 4 times the average dose to be lethal to a human. The therapeutic index is a measure of a drug's safety margin. The higher the value, the safer the drug. This second example from your text shows a very narrow margin of safety with a therapeutic index of only 2. Here is a brief list of some of the more common narrow therapeutic index drugs. So in order to make sure that patients aren't receiving toxic levels of these drugs, we need to monitor the plasma concentration of those drugs to see if they're not at toxic levels. Number 2, we can look at the biological effect of the drug. So for example, if I'm giving warfarin, I'm going to monitor the international normalized ratio, or INR, to make sure that the plasma viscosity is where it needs to be. A third way to monitor drugs is its clinical effect. So if I'm giving an antihypertensive, is it in fact lowering the blood pressure? So careful monitoring and vigilance is important with these drugs. This is a graded dose response curve. It allows us to graphically visualize an individual's therapeutic response at different doses of a drug. There are 3 phases. Phase 1 occurs at the lowest dose when few target cells have been affected by the drug. Phase 2 is the straight or linear line of the curve, which demonstrates the relationship between the amount of drug administered and the degree of client response. This is the most desirable range. The more drug you administer, the greater the response. And finally, phase 3. A plateau is reached for several reasons. One reason is that all of the receptors for that drug are occupied, or that the drug was successful in its desired outcome, such as 100% relief from pain. Increasing the dose, therefore, cannot possibly produce any more relief or therapeutic effect. However, increasing the dose at this stage could produce adverse effects. Now, all drugs within a class are not created equal. Some antihypertensives, for example, are better at lowering blood pressure than other antihypertensives. Some analgesics are better at targeting pain compared with other analgesics. Furthermore, drugs within the same class are also effective at different doses. So how do we compare drugs within a therapeutic or pharmacological class for their effectiveness so that we can provide treatment? Well, there are 2 ways of doing this. First, we examine the potency of a drug. The more potent the drug, the lower the dose required. The second method is efficacy, which in simplest terms is the drug's ability to get the job done. What is essentially the maximal response you can get out of a drug? That's efficacy. If I have a headache, I can choose between ibuprofen, which is 200 milligrams, or I can take acetylsalic acid, which would amount to about 650 milligrams. They are both analgesics, but which one is more potent? Right, the ibuprofen, because I need a smaller dose compared to the acetylsalic acid. Well, now, both would effectively get rid of my headache. Therefore, which one is better efficacy? Well, both are the same. If they both get the job done, then they have the same efficacy. What if, however, I have a severe blinding migraine? Would ibuprofen or acetylsalic acid get the job done? Probably none. I would need something stronger, such as a narcotic within the analgesia drug class. When it comes to pharmacotherapeutics, what do you think matters most to the client? The drug dose or effective treatment? Realistically, I'm not concerned about the dose if I'm that sick. I'm concerned about pain relief. So, what does a drug do? How does it do what it does? When I take a pill, what happens at the molecular level? Well, let's go closer, deep down into our bodies, and have a look at the cellular receptors and the drug action. Cell signaling is basically how a cell perceives and responds to its environment, and how it carries out its basic cellular activities. I make that sound simple, but it's far more complex in reality. Cells get their information from macromolecules known as receptors. It is to these receptors that drugs bind and initiate their effects. Many of these receptors are cell surface proteins, so they're sitting on the surface of the cell. But some of them are actually found inside the cell. Molecules that activate, or in some cases inhibit, these receptors are called ligands, such as drugs. It is important to know that receptors don't exist just to bind to drugs. They bind to endogenous molecules that are also ligands, such as hormones, neurotransmitters, and growth factors. Endogenous, meaning it's originating from within the body, unlike drugs that are introduced from the body from outside of itself. The amazing concept of a drug binding to a receptor and causing change in body chemistry and physiology is incredible, and it's the fundamental principles of pharmacology. To even affect change, it means that this foreign drug needs to be able to interact with molecules and chemicals that are found in the body. A drug attaches to its receptor much like a lock and key. It either fits or it doesn't. Once bound, drugs trigger a series of second messenger events inside the cell. These second messengers relay and amplify signals received from the ligand or drug on the cell surface to target molecules in the cytosol or nucleus, initiating the drug's action to either stimulate or inhibit the cell's normal activity. The underlying biochemical mechanism of drug-receptor interactions is very complex, but simply put, either the cell's activities are enhanced or they're inhibited. When a drug binds to its receptor, it can produce a response that mimics the effects of an endogenous regulatory molecule. There are three ways it can do this. One is known as an agonist. These are drugs that bind to a receptor and produce the same response as an endogenous chemical. Sometimes agonists can produce greater maximal response than the endogenous chemical. Second, we have partial agonists. These drugs also bind to a receptor but produce a weaker response than an agonist. And then finally, we have antagonists. They occupy receptors, thus preventing endogenous chemicals from acting. They compete with agonists for receptors. Other antagonists inhibit the effects of agonists by changing pharmacokinetic factors. So for example, there's drugs that might slow the absorption of a drug. So that's as far as we're going to go into pharmacodynamics in this course. I've highlighted some very important parts for you to keep in mind as we move forward in this course. The first that's very important is that you realize how different people are and how so very different we respond to medication. These are known as idiosyncratic responses. So based on those curves that we looked, we could see that the majority of people might respond to an effective dose. But some people might get toxicity from the same dose or need a higher dose to get the same response. How we respond to drugs also has to do with the potency and efficacy of a drug as well. We've also looked at cellular interactions with receptors and ligands, and it's important for you to know what exactly an agonist, a partial agonist, and an antagonist and how that functions with the cell receptors. The future of drug therapy is full of amazing possibilities. When we used to think that one drug was a one-size-fits-all for people, thanks to the Human Genome Project and other advances in pharmacogenetics, we might even be able to customize drugs for unique individuals. So thanks for joining me. I'll see you all in class.